So the idea that the neuron is a one or a zero and giving rise to bit-like states emerging into consciousness is incredibly insulting to biology and to anybody who really thinks about it. Despite that, some of our leading thinkers do think that, which makes me think that they're getting so much money, no offense, from AI that they're selling out and saying, sure, AI can be conscious, and all the cartoon neuron labs all over the world are getting rich because AI is pouring money at them to keep promulgating the same bullshit.
Hi, I'm Craig Smith, and this is Eye on AI. Today, I have a fascinating conversation with Stuart Hameroff, an anesthesiologist who, with physicist Roger Penrose, proposed a theory of consciousness that suggests quantum processes within the brain play a fundamental role in generating conscious experience. Stuart's work as an anesthesiologist, turning on and off consciousness, provides him with a unique perspective on how consciousness works. We delve into his ideas about how quantum effects within microtubules in neurons in the brain might lead to conscious awareness. I hope you find the conversation as thought-provoking as I did. First, however, I need to give a shout out to our sponsor, NetSuite by Oracle. So here we go. What does the future hold for business? Ask 9 experts and get 10 answers. Bull market, bear market, rates rising or falling, inflation going up or down. Can somebody please invent a crystal ball? Until then, over 40,000 enterprises have future-proofed their business with NetSuite by Oracle, the #1 cloud ERP, bringing accounting, financial management, inventory, HR into one fluid platform. With one unified business management suite, there's one source of truth, giving you the visibility and control you need to make quick decisions. With real-time insights and forecasting, you're peering into the future with actionable data. If I were a larger organization, this is the product I'd use. Whether your company is earning millions or even hundreds of millions, NetSuite helps you respond to immediate challenges and seize your biggest opportunities. Speaking of opportunities, download the CFO's Guide to AI and Machine Learning at netsuite.com/eyeonai. That's NetSuite, netsuite.com/eyeonai, EYEONAI, all run together, to get the CFO's Guide to AI and Machine Learning. The guide is free to you at netsuite.com/eyeonai, netsuite.com/eyeonai. There's a lot of talk about the potential for sentience in large systems, which devolves to the question of, dualism versus materialism, whether or not the consciousness is an emergent property. I mean, you are deep in all this stuff. But this quantum effect just sounds like an interesting way to approach it. And I know it's controversial and there are people that don't buy into it. But I thought for listeners, could you start by giving your background? And I wanted to talk a little bit first about anesthesia, because a lot of people don't realize that anesthesia is not simply putting someone to sleep. It's cutting off consciousness. And talk a little bit about how you came to understand or how view consciousness through your work in anesthesia, and then we'll talk about microtubules and quantum effects and all of that. Is that okay?
Well, most people view the brain as a complex computer of simple neurons, and this lends itself to computers, to AI. And this goes back to the Hodgkin-Huxley neuron, 1950s, where it was modeled, the neuron was modeled as an integrate and fire threshold logic device, no different from a threshold logic device in silicon. Algorithmic, operating at one fairly low frequency in hertz, under 100 hertz. And inputs are integrated in the dendrites and soma to a threshold, and an output is fired, and that's no different from a threshold logic device you could make a computer out of. So the idea became that the brain is a computer of neurons connected by variable strength synapses, and that neurons are no different than and the firings are the ones or the zeros, the bits. So you can make a nice computer out of that. And that's what people have done, and assumed that that's how the brain works, and assumed that since that's no different from AI, that AI can therefore be conscious, since the neurons, the bits in AI are functionally or in any way no different than brain neurons. That's bullshit. Okay. It's bullshit because neurons are alive and we don't yet know what that means. And, there was this vitalism back in the 18th, 19th century, and that fell by the wayside. But quantum vitalism is a very valid principle put forth by Schrodinger. He said that periodic lattices or aperiodic lattices could have coherent vibrations, coherent oscillations, coherent states, including for the unity of binding and that sort of thing. So he raised the possibility of quantum coherence as a quantum vitalism that would differ neurons from inanimate objects in such a way that could explain consciousness. Although he didn't actually say that. So the point is that I think this whole AI, brain equals mind equals computer, based on and this is what Roger was fighting against back in 1989. The idea that AI was automatically was no computation and consciousness were the same thing, and Marvin Minsky was the big guy then, and I think he's the emperor in the Emperor's New Mind, and basically the point was the emperor has no clothes, the emperor has no mind, because consciousness is not a computation, and he argued that the first part of the book from Gödel's theorem, which basically said that a mathematical theorem cannot prove itself. It must be judged to be true by an outside observer, something outside the system, like a conscious mathematician. And John Searle had said a similar thing with this Chinese room argument. You could do all kinds of sorting and classification without any understanding. Yeah, so Roger applied that principle to understanding. Basically, the mathematician understands the equation is true or false, or the theorem is true or false, and understanding means that we can judge a value of something, and he likened understanding to a mathematical proof, which meant that consciousness required something outside the immediate computational system, which is a classical system in the brain, the brain-mind computer, where the neurons are like bits. So something else was required, something outside the system. And the second part of the book was where he looked for that something outside the system. The only thing outside the classical world is the quantum world. And so he looked for it in quantum physics, in the big remaining mystery in quantum mechanics, the measurement problem, which is to say that we know there are superpositions at small scales and quantum scales. We know there are quantum superpositions where particles or things can be in multiple states or locations at the same time. And yet when we observe or measure them, we only see one definite state. So we go from the quantum multiple possibilities to the classical one definite state, possibly through the very act of observation or measurement. But if it's consciousness, that puts consciousness outside science, and that is a dualist position. If you say that you need consciousness to observe a quantum superposition and cause the collapse, that's putting consciousness out. That's dualist. You can't explain consciousness, but you can use it, or they try to use it to explain the measurement problem. So the other interpretation of the measurement problem, as you may know, are multiple worlds and decoherence, and then Roger's view. So, but Roger started out and did something nobody else has done that I'm aware of, which is try to explain superposition. How can something be in multiple places or states at the same time. And his answer was to go to general relativity, Einstein's general relativity, where matter was equivalent to curvature in space-time. So this is for very big things like the sun, and he said that stars behind the sun, the light would be curved, and then come around and could be seen on Earth. And Eddington did that experiment in an eclipse in 1919, and saw stars known to be behind the sun, proving that space-time curved the sunlight, the starlight, proving Einstein's theory of general relativity that space-time curvature was
Yep.
equivalent to mass. So Penrose applied that to very tiny things. Okay, so Einstein did it for big things. Roger said, well, why not quantum particles? So if you have a quantum particle, it's here. It has a tiny curvature. And if it's in two places, that is a curvature this way and a curvature this way. And if you think of space-time, well, the easiest way is to think of it as a two-dimensional sheet, just condensing it down to 2 dimensions. You can think of it as a sheet, and so you have a curve one way, curve the other way, and you have a separation in space-time geometry. So superposition is you have a curve here and a curve here. And you can imagine that if those separated curvatures continued, that each one would form its own universe, and you'd have many worlds. So this superposition separation lends itself to quantum physics and general relativity, and you can at least picture how something can be in multiple states at the same time. But Roger said that these separations in spacetime were unstable, and after a time t equals h bar over E sub g, where E sub g is the amount of superposition mass where spacetime is separated from itself, which you can calculate. The larger that, the faster it'll collapse. And rather than consciousness causing this collapse itself occurred spontaneously at that equation and emitted, corresponded with a moment of conscious awareness, of experience. So this was the source of consciousness.
Mhm.
Rather than consciousness causing collapse occurred spontaneously due to quantum gravity or general relativity and gave rise to consciousness, making consciousness a fundamental property of the universe, which a lot of people say in different ways, like in panpsychism or this or that. But this was a very specific way that explained the measurement problem in quantum mechanics and the origin of consciousness and unified general relativity and quantum physics. So 3 for the price of 1.
Hmm.
And this has actually been criticized by people like David Chalmers, who says things like that we, counting me when we applied the microtubules, we are invoking a mythical law of minimization of mysteries, using one mystery to solve another one. And I say, why the fuck not? Pardon my language. Occam's razor would favor one solution for 3 mysteries. Why not? I mean, why is that even a criticism? It should be like, hey, that's pretty cool. Anyway, Chalmers and many others have been very disapproving of all this, and for no good reason, except I don't want to speculate why, but I think they're wrong, and I think we're right, and I think the evidence is on our side. So in the meantime, Chalmers and Christoph Koch and all the big shots in neuroscience, the neuroscience of consciousness at least, have led the field into a hole, a dead end, where these I call them cartoon neurons because they're only surface, they're algorithmic, they're not biological, they're cartoons. And you can make a computer of cartoon neurons, but that's not consciousness. But it sucks up to AI, and I know you're an AI advocate, and your listeners probably are too. It sucks up to the idea that that AI can be conscious. And I mean, AI is great. When I just retired from anesthesia, we had AI systems in our anesthesia equipment, fantastic,
Yep.
But that doesn't make them conscious. And I resent the hell out of the fact that people are claiming they're conscious based on cartoon neurons, which have absolutely no bearing on what's going on in the brain. So, okay, if the brain's not a computer with consciousness emerging at a high level of complex computation, which I don't think it is, what is it? So, one way to look at it is, okay, neurons are alive. Let's look at their cells. let's look at a cell like a paramecium. It's one cell,
Mhm.
no synapse, no network, just one cell. Swims around, finds food, finds a mate, has sex with a mate, avoids obstacles, can learn, and it does it with its microtubules in cilia and inside the cell that process information. The same structures are found in our neurons, exact same structures, and they organize the interior of neurons. And if a paramecium can get laid, why is a neuron a one or off switch, one or a zero? It's an insult to neurons. I mean, the slime mold amoeba is one cell that oozes, and people have made these tests where the slime mold, by putting aversive or attractive things in different places, can solve the traveling salesman problem. And I
Mm.
give you a reference for that. So, and it does it by its microtubules picking out where to go. So the idea that the neuron is a one or a zero and giving rise to bit-like states emerging into consciousness is incredibly insulting to biology and to anybody who really thinks about it. Despite that, some of our leading thinkers do think that, which makes me think that they're getting so much money, no offense, from AI, that they're selling out and saying, sure, AI can be conscious, and all the cartoon neuron labs all over the world are getting rich because AI is pouring money at them to keep promulgating the same bullshit. No offense.
Who is the head of the Allen Institute now who just changed his view on
Christoph Koch.
Okay, Christoph Koch, yeah.
He's no longer the head. He stepped down. He's just a scientist there.
Okay, but he
And
changed his view from sort of purely materialistic to something
mystical.
mystical. I don't quite understand
And
it.
he
This
skipped over
universal.
he skipped over the quantum stuff, which could have made it possibly true.
Yeah.
And so he's trying to have it both ways. He's trying to be mystical and panpsychist and other forms of consciousness altering things, and yet he sticks to the goddamn cartoon neurons. In fact, Christoph and his collaborator, previous collaborator, Francis Crick, are responsible for the cartoon neuron. In fact, Crick, DNA, all that, wrote a book called The Astonishing Hypothesis. So
Right.
Crick wrote this book, The Astonishing Hypothesis, and the astonishing hypothesis was that the brain is nothing more than a pack of neurons acting like bits in a computer. So that was astonishing. I thought it was stupid, but that was his claim, that it's nothing more than that. There's nothing magical, nothing mystical, nothing, you know. And then so he worked with Christoph. Christoph was a young upstart at Caltech, and they started hanging out together and started writing papers and stuff together. And Christoph, despite being supposedly the world's leading neuroscientist, became a cartoon neuron advocate. And I know that because I organized the Science of Consciousness conference in 1994 in Tucson.
Hmm.
And we just had the 30th anniversary last April. And at the very first one, I invited both Chalmers and Christoph. Chalmers was a completely unknown postdoc philosopher who lobbied his way onto giving a plenary by sending me faxes. I can't remember. Maybe we were just getting email then. And he had a pretty interesting abstract. And the first digress a little bit. The first morning was philosophy, and the first two speakers were well-known philosophers who gave the most boring talks you can imagine. Back then, philosophers didn't use slides; they read a paper, no eye contact. It was incredibly boring. So we had an interdisciplinary audience, and after the first two talks, everybody's snoring and looking around. And then Dave hopped up, and I had, like I said, he had lobbied me into giving him a plenary, and I'm glad because he gave a fantastic talk, the famous hard problem talk. He goes, we neuroscience, we have all these things like memory, attention, behavior, reportability, verbal, blah, 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 blah. As hard as they are, they're easy compared to the nature of experience. Why do we have qualia? Why do you have the color yellow, the pink, the feeling of joy, of envy, blah, blah, blah. So he basically announced the hard problem. and it worked. The audience got it. I mean, there were all these people from all different walks of science and life and this and that. And oh, the heart problem. That's why we're here. Who cares about all this
Yeah.
other stuff? Let's talk about consciousness. So he identified the heart problem, and he's, it's gonna. He said basically it's gonna probably take a really crazy theory to explain it. Most theories won't be crazy enough. And then later he said our theory is too crazy, but that's beside the point. So anyway, that kind of galvanized the conference and the movement. So that was one famous talk. Then Christoph gave a talk, and I had invited him, and he said, "Well, is this like a woo-woo conference, or is this real science?" And blah blah blah. I go, "No, this is real. We're gonna have all kinds of people." So anyway, he showed up, and he gave a talk, and he talked about his work with Crick, and he showed the neuron, his model neuron, where it was basically the Hodgkin-Huxley neuron, where all the signaling was on the surface membrane, and it was algorithmic and linear. You reach a threshold and you fire, and the firing is a bit or a one or a zero. And I was chairing the session, and so I took the first question. I said, Christoph, why don't you look inside the neuron for stuff that could process information that might contribute to consciousness? And he said, Stu, I'm going to answer you in the persona of my favorite movie actor, Clint Eastwood. And Christoph's a character. He does shit like this. So he said, and it turns out it's a line from Magnum Force. I found out later. A man's got to know his limitations. That was his response to why he didn't look inside the neuron. And everybody laughed, and he's being a movie actor, and I kind of took a step back. I said, "Well, I didn't expect that," and I didn't really have a snappy comeback, so he got away with it. He got away with planting the seed of a cartoon neuron that has persisted for 30 years in the science of consciousness. So when I hear that serious neuroscientists believe in cartoon neurons, I say that's an oxymoron. A serious scientist really cannot believe in cartoon neurons. But okay, why not? So now it's on me to explain why you can't. And for one thing, for 20 years, so I got interested in microtubules when I was in med school. I was in consciousness, but studied cancer and cell division, mitosis. And the microtubules pull the chromosomes apart. And if they don't do it perfectly, you get abnormal genotypes, phenotypes, and cancer and all kinds of bad stuff. They have to work perfectly to get the exact matched pair. Everybody else in the lab got into the chromosomes. It was the dawn of the genetic revolution. But I got interested in the microtubules. How the heck do they know which chromosome to pick, where to go, what to do, what's organizing them? And right then, this is all in the early 70s. And within a couple of years, I personally discovered, well, I didn't discover them, but I learned about microtubules. I was interested in consciousness, started to think they might have some information, intelligence, or maybe consciousness. Who the heck knows? And their structure became known by X-ray crystallography, and it was a cylindrical lattice with cylindrical hexagonal lattice with Fibonacci geometry. So that if you followed the helical windings, you got Fibonacci mathematics and geometry. And I thought that was pretty cool. And they were everywhere. It was also discovered back then that neurons were full of them. And ironically, for 30 years prior to that, the fixative agent for the electron microscope had been dissolving the cytoskeleton, so that the electron microscope was showing the picture of a cell as like a minestrone soup of stuff swimming around. When actually, when they switched fixative agents, they either went from osmium tetroxide to glutaraldehyde or the other way. I think that's the way it was. All of a sudden, they saw all this structure. It was a guy named Keith Porter at Harvard, a famous electron microscopist, switched fixative agents. All of a sudden, saw all the structure inside cells, inside neurons. Neurons, being the most asymmetrical, had the most extensive cytoskeleton, microtubules, because you need structure to maintain asymmetry. So within 6 months or so, I learned about microtubules functioned and looked like computers. They were everywhere in the brain, particularly the brain, and they had the potential to process information. So I I knew engineers and physicists who were interested in like the game of life and cellular automata and information processing and that sort of thing. People from Los Alamos. I met these nonlinear dynamics people, including Alwyn Scott, who started the Center for Nonlinear Studies at Los Alamos. And through him, I met other people, including a good friend of mine, Steen Rasmussen, who's a physicist in Denmark. And they invited me to these meetings at Los Alamos, which wasn't very far from Tucson. So I went to like chaos meetings, cellular automata meetings, this neural net, this and that. And cellular automata, in fact, they invited me prior to that, they invited me to give a talk in the math department. And I was talking about information processing and microtubules, where I had this model of each tubulin could be like a 1 or a 0 interacting with neighbors in this lattice. And somebody said, "Are you describing a cellular automaton?" And I go, "What's a cellular automaton?" And they said, like the game of life. I'm going, "What's the game of life?" I mean, I was an anesthesiologist talking to a bunch of mathematicians. So I soon found out, and I soon found myself at Los Alamos at a cellular automata meeting, presenting a model of microtubules as cellular automata, the cell being the individual tubulin. So cellular normally refers to a cell, but cell is already too big. So automata function. But we needed, so working with the physicists, we needed a clocking mechanism, because in cellular automata, they update on every beat of the frequency. And so all the interactions occur at the same time. And there was a model called Frohlich coherence by this guy Herbert Frohlich, who had been a quantum guy who got into biology and postulated that room temperature biology would have quantum coherent oscillations due to nonpolar areas. So this was key, because in biology, everybody says the brain's too warm, wet, and noisy for delicate quantum effects. And it's true, biology in general is 70% water. But the other 30% includes all kinds of other stuff, including the microtubules, which, if you go inside any protein, including microtubules, the polar stuff, the charge, the water soluble stuff is on the outside interacting with the water. But the oil-like nonpolar organic molecules, like benzene, I mean, we're talking about organic chemistry. The basis of life is all built around the benzene and indole rings with these pi electron resonance. And they coalesce in the middle of the protein to get away from the water. And in microtubules, they form these big regions that connect with the region in the next tubule, next one. So you have these nonpolar regions which are quantum friendly, which support quantum effects like quantum optics, fluorescence, superradiance, and so forth, which run the length of the microtubule. So it's already a mesoscopic or macroscopic quantum state, which is exactly what you need. So the idea that the brain's too warm, wet, and noisy, it's not noisy when it's coherent. So Frohlich had the idea that these were all oscillating coherently, kind of like a laser. And so depending on the mass, it could be in megahertz, gigahertz, or terahertz. And so we modeled them, just borrow that, mathematically model the microtubule processing information, where it's switching and updating every Frohlich coherent period, which could be, let's say, megahertz. So that was our model. And people. So I was going around to neural net and AI meetings, being a pain in the ass, because I was saying, like the singularity. Well, it started with Hans Moravec, and I read his book in 1986, Mind Children, where he said, okay, there's a 10 to the 11th neurons switching about 1,000 synapses. So that's 10 to the 14th at about 100 hertz. So that's 10 to the 16th operations per second for the brain. Mm-hmm. And then Kurzweil picked up on this for the singularity, 10 to the 16th. When we get to 10 to the 16th operations per second, we'll have brain equivalents and consciousness in a computer, and just give us a few more billion and you can download your consciousness, all these ridiculous promises. But I was saying, hold on, fellas. You actually have about 1 billion tubulins in every neuron switching at 10 megahertz. That gives you 10 to the 16th operations per neuron per second, multiplied times 10 to the 11th. So I wasn't very popular in AI and still aren't, probably. And because because I was saying that, your goalpost is way, way, way downstream, just based on information processing. And so they didn't like that. And but then one day, fortunately, somebody said to me, and I wish I could remember who it was because I owe him a debt of gratitude. He said, "Okay, let's say you're right. How would that explain consciousness? How would that explain love, joy, feelings, envy, blah, blah, blah?" And I was a little bit stunned. I had no idea. This was the hard problem being thrown in my face 4 years before I heard Dave give the hard problem talk, or before he gave the hard talk. I mean, the idea had been around a long time.
Yeah.
But Dave articulated it very well, and this guy articulated pretty well, and I got it immediately. I said, "Holy shit, I'm a reductionist. What am I going to do now?" Because I didn't think that was very promising. Fortunately, that same individual suggested I read The Emperor's New Mind by Roger Penrose, and so I did, and I didn't understand it. It's so dense. You saw it's really hard to follow,
Yeah.
but I kind of looked at the pictures and I gleaned a little here, a little there. It was obvious the guy's a freaking genius, and I knew it seemed like everything about everything. And if only I could, understand a glimpse of it. So I got the non-computability. Okay, I was sold on that. I had heard Searle came to Tucson a few years before and talked on the Chinese room argument, which convinced me that there's something more to it than the computation. But Roger needed this outside influence, which was this quantum stuff, which I had never really tried to understand, but I did. I did my best, and I realized at the end he said, "Well, we need some kind of quantum device in the brain that can be in a superposition of 1 and 0, and then reach threshold. It has to be pretty small, so neurons are already too big." But he didn't know what it was.
Yeah.
And so I read the book and I said, "Holy crap! He's talking about he needs microtubules. This is exactly, they're there in every neuron. They control the neuron. The quantum stuff inside the Frohlich, it's quantum. It's exactly what he's looking for." So I wrote him a letter, old-fashioned letter, by post, and mailed it. I told him I was going to be in England for a different conference in a few months hence. I'd be happy to visit him. And I got a letter back. He said, "Yes, please come to Oxford and we'll talk." So I did, and I met him on a fateful day, and he met me at the train station. We went to his office, the Mathematical Institute, and he said that he was going to a conference on consciousness at Cambridge coming up with Pat Churchland and Dan Dennett, two materialist reductionist philosophers. And I said, "Oh, that sounds fun. I'd like to go, but I have to go to this other conference. I have to give a talk." So anyway, he said, "Well, tell me about microtubules." So I did most of the talking. I just started talking, and he was really taken by the Fibonacci. That was Fibonacci geometry. He asked a lot of questions. First question was, "These things are real? They're not like simulations of something?" I said, No. They're found in every cell. They're very important, blah, blah, blah. So like I said, I did most of the talking. And at the end, he said, "Well, thank you very much. That was really, really interesting." And, I gave him some papers and I showed him a book I had written on microtubules, and shook his hand and said goodbye. And I walked away and I said, "Wow, that was cool. I got to meet Roger Penrose. He's a really, really interesting guy." So I went to my meeting. I think it was, where was it, Brighton or someplace. And 2 weeks later, I was back in London and I was having dinner with a friend. He said, "Hey, I went to this meeting in Cambridge and Roger Penrose was talking about you and your stupid microtubules." And that was a thrill. I got to admit, that was a real thrill. And soon afterwards, I got an invitation to a meeting in Sweden. No audience, just scientists, including Roger, Dan Dennett, a few others, north of the Arctic Circle in an isolated research station. So I said, absolutely. So I went and I met Roger and his wife, and we spent 4 or 5 days out there playing a lot of ping pong, hiking at midnight, skiing. We went skiing at midnight, and just started talking about microtubules and quantum and how we could develop a model. And then we met a few about a month later for 2 weeks in Denmark, where I had spent my sabbatical, and he and his wife always went there to go to their dentist, spent 2 weeks a year there. So we spent some time there together. In fact, I was with him at lunch there when he had somebody come up and said, Roger, you just became knighted.
Yeah.
And at first I heard of that. I mean, he said, oh, yeah. I said, wow, you're going to get to meet the queen? He says, well, I've met her several times. She's very nice. He's very nonplussed about things.
Sure.
He didn't want to fuss about it. He was kind of embarrassed. Same thing when he won his Nobel Prize years later. But anyway, so we started coming up with a theory of how we could quantify based on the simple equation. Collapse occurs at time t equals h bar over E sub g, where we could calculate E sub g for tubulins and figure out how many tubulins we needed in superposition to reach collapse for a conscious moment at certain frequencies. So Vanessa, his wife, said, I encourage you to work with him, but I just have to warn you, he's very meticulous. He's very slow. He's doing 20 things at once. This will take a long, long, long time. So be patient. I said, thank you. That is good to know, because I'm easily frustrated by things like that. But it was such a thrill to work with Roger. So anyway, 3 or 4 months later, we still don't have a manuscript, a rough draft. We just have some general concepts and how to quantify this and that. They're making progress, but nothing close to a manuscript. And we got a letter from, or an email, I guess, from the publisher of the Journal of Consciousness Studies, who said that Pat Churchland, and her graduate student, had written a preemptive strike against our theory before we'd even published on it. And if we could write a response in the next issue, but they needed a manuscript in 2 weeks. And I'm thinking, holy crap, it's been a year. We don't have a manuscript. How are we going to come up with a... So I talked to Roger about it, and he was sufficiently annoyed by it, as well he should be, that we... So basically, there were 2 parts of their attack. Number 1 was on Gödel's theorem, and whether that was applicable, and Hillary Putnam, and all these arguments back and forth that I didn't understand. I didn't have a freaking clue. The second part was about microtubules. And the main argument they had was that there's a drug called colchicine, which is used for gout. So in gout, you get uric acid crystals in your joints, quite often the large toe, but it can be any joint. And the immune cells, the lymphocytes and macrophages, move into the joints and then release all these bradykinin, which cause this inflammation, swelling in enclosed space, and it hurts like hell. So the colchicine, so the lymphocytes move by the microtubules assembling in a certain direction, then flowing, and then going this way, that way. So that's how they move. So colchicine prevents microtubules that are assembled from disassembling and then reassembling. So it does inhibit this movement of these lymphocytes and macrophages. Therefore, it treats gout. It's good for it. And they said, but people who take colchicine don't lose consciousness. So consciousness cannot depend on microtubules. So I said, Roger, I got this one. So I said, number 1, colchicine only affects microtubules that are actively disassembling and reassembling. The microtubules in brain cells, neurons, don't have to do that because they don't have to, nerve cells don't divide. So they don't have to disassemble for mitosis and then reassemble, in which case you'd lose any information you had stored in the lattice. And they affect lymphocytes because they need to do that to move. But again, you can screw up the lattice, but it doesn't cost you anything because you're not storing any memory there. In dendrites and soma, neurons, the microtubules are capped at both ends. So you can't add at one end and lose the other, which is called treadmilling. Mm-hmm. The neurons don't divide, so they don't disassemble so they can reform as mitotic spindles. They just kind of get hardwired, and they're perfect for storing memory, actually. And in fact, that's where I think memory is stored. So I said, okay, so the colchicine, number 1, it doesn't cross the blood-brain barrier, so it couldn't affect brain microtubules anyway. Number 2, it only affects microtubules that are disassembling and reassembling. So even if it got there, it wouldn't do anything to them. And number 3, I found a paper where somebody, for some reason, had injected colchicine into the brain of rats and wiped them out. They were like brain dead. And so that was my part, a few other things. And so we published two, and that was our first paper. And the title of their paper, and I don't know if you know Pat Churchland, she's still around, she's very old, really caustic, annoying, sarcastic. And she's in the beginning of their paper. Well, first of all, they call, Roger's famous for Penrose tilings, tiling a plane with self-repeating patterns that don't repeat and that sort of thing. And so the title of their paper was "Gaps in Penrose's Toilings," as a pun on Penrose's tilings.
Right.
So there was a gap in his toilings on consciousness and blah blah blah. And in like the first or second sentence, they said this theory is no better supported than 1 in a gazillion caterpillar with hookah hypotheses. So that was their opening line. Welcome to consciousness. We're gonna fuck you up big time. And so they started off with saying we were from Alice in Wonderland and whatever that implied. So at the end of our paper, we answered all the points. At the end, I said, Roger, we have to say something about the stupid Alice in Wonderland thing. And I said, how about we say, it's not that we're in Wonderland. Perhaps their heads are in the sand, like ostriches.
Yeah.
And Roger said, oh, yeah, that's fine. But let's make it Shakespearean, praps, like P apostrophe R-A-P-S. I said, fine. So that's what it was. So then we sent it in, and then Roger and I, we were at a meeting at the Fetzer Institute, actually, and we get a fax from the publisher. They're about to go to press, and he says, I commissioned a cartoonist to draw a picture of this dispute between you guys and Pat and Grush, and I want to use it, but I want your permission to use it. I said, okay. I said, well, Roger and I together faxed it to us, and it's a picture of so I told you the caterpillar with hookah stories. So it's a picture of in the middle are 2 giant ostriches with their heads buried in the sand and their butts pointing out at the camera, the viewer, saying G for Grush and C for Churchland. And on the left is a big mushroom with a caterpillar sitting on top of it, smoking a hookah. And the caterpillar looked suspiciously like me. In fact, it looked exactly like me. And I was blowing the hookah smoke up the butts of the 2 ostriches. And then on the other side, Roger was dressed as the White Rabbit, holding the keys to the Platonic realm behind him. So that was the cartoon. I said, that's fine with me. I don't know if Roger will go for that. And Roger looked at it and he chuckled. He said, okay, fine. So that got published, and I could dig it out if it's really funny.
Yeah. I'd love to see it.
Okay.
So before you go on, can you? Well, where do you want to go from here? Do you want to explain how you guys think consciousness arises from these quantum effects in the microtubules, or I'm interested in the nature of consciousness and anesthesiology, like
right.
So I'll let you go from there.
Okay. Well, let me give you a little more background, and I'll get to that. Because I told you we were relying on this theoretical Frohlich coherence, but then in about 2010, this guy, an Indian guy, who becomes a key figure in this whole story. His name is Anirban Bandyopadhyay, and he was a grad student or a postdoc in India, and he won this international prize from the National Institute of Material Sciences in Tsukuba, Japan. The number one Japanese material science institute, the top 3 in the world, to have his own lab. So he didn't have to apply for funding, no postdoc, just here's a lab, here's money, do what you're going to do. And they gave 3 of them per year. So the first thing he did was build a molecular computer out of something that was published in Nature or something. And then somewhere along the line, he read about me and Roger and microtubules, got interested in microtubules. And he started studying them and doing nanotechnology. And to make a long story short, he found that microtubules have coherent oscillations that repeat every 3 orders of magnitude. going down in frequency from kilohertz to megahertz. Sorry, going up in frequency, down in scale. Kilohertz, megahertz, gigahertz, terahertz in microtubules. And at each level, they have a coherent resonance pattern that's exactly the same, a triplet of triplets. And so he did this at 3 scales. He started with, like for one microtubule, he attached 10 nanoprobes and used a couple of them to stimulate. And if you stimulate a microtubule with a voltage, it's an insulator. But if you stimulate it and alternate the voltage or use AC, you'll find certain frequencies where there's a huge conductivity. And then it goes along, and then 3 orders of magnitude later, you see the same huge conductivity. So every 3 orders of magnitude, you see this resonance conductivity with a particular pattern that is a triplet of triplets. You see 3 peaks, then each peak has 3 peaks. And he found that in kilohertz, megahertz, gigahertz, and terahertz, and arguably you could say the EEG in hertz is a triplet of triplets or something like that. And so that was evidence, number one, for the coherent vibrations we needed for just automata function. But some of these frequencies were in the quantum realm, so it got us into the quantum realm and got around the problem of the brain's too warm, wet, and noisy for quantum effects. And but Anirban, he had this further theory that this actually went further down into the fine scale structure of the universe, that these triplet of triplets continued in space-time geometry all the way down to the Planck scale. And this is very similar to Roger's idea that somehow these quantum processes were reaching the Planck scale or were rooted in the Planck scale. So Anirban had this model, and I've come to adopt it, actually, that you have these triplets going down, and once you get faster than terahertz or petahertz, you're smaller than biology, and it can continue somehow in space-time geometry, which other people have said had fractal properties as well. So Anirban calls it a great chain, a great resonance chain, or a scale invariant hierarchy, but I think in the brain, at least, this goes. If you go inside neurons, you see this hierarchy going smaller, deeper, faster more into the quantum realm. Now, a lot of people have had hierarchical models in the brain, starting from the level of neurons as the fundamental units, going up. So you have neurons, networks of neurons, networks of networks of neurons, getting larger and slower. And a lot of people have had these kinds of hierarchies. But if you go the other way, into the neuron, into the microtubules, you get smaller, faster, deeper, and quantum stuff. And Anirban calls the microtubule behavior time crystals. So a time crystal was described by Frank Wilczek, who won a Nobel Prize for one of the nuclear forces, I forget. And he came up with the idea of a time crystal, which is actually a kind of a perpetual motion machine, but because the ground state of it is an oscillator. But that can happen because depending on what the ground state is. I mean, there's energy at the universe, there's energy at the Planck scale, there's energy in biology at KT. So, but the point of a time crystal is that in a spatial crystal, the spatial pattern repeats wherever you look in the crystal. In a time crystal, the same pattern appears at different frequencies. Mm-hmm. So in a microtubule, it had the exact same resonance frequency pattern in kilohertz, megahertz, gigahertz, and terahertz, and probably petahertz. And that might go all the way down. We've actually, in our astrobiology work, we found suggestion of same sort of thing in molecules from a meteorite. These are polyorganic molecules. We're looking for the origin of life and consciousness. I retired from anesthesia, and I'm working in astrobiology now with a guy named Dante Loretta, who brought back these samples from the asteroid Bennu. They just got back last September. So we're going to be looking at these organic molecules. So this time crystal behavior might be a kind of a sign of life, or maybe even consciousness. We think that actually consciousness was there at the beginning and prompted the origin of life, because otherwise there's really no reason for all that purposeful behavior before genes and brains. So we think consciousness was there right from the start, and with Roger's mechanism, it could be. But that's kind of a somewhat of a digression, but I think a very important one. But going back to where we are now, so rather than a cartoon neuron of a neuron either firing or not, we have this multi-scale hierarchy. And and you could say, well, this is reductionist because you're going into one neuron. But when you get into the quantum, these microtubules in that neuron are entangled with the ones in the next door and next door, next door. So you can have big parts of the brain entangled into one fundamental quantum unit, which is what you need for conscious, for binding, for example, for unity of conscious self. So the Orch OR events that happen are specific events, but they're entangled, and so they're brain wide, and they happen at a particular frequency, and they're actually too fast for cognition because to avoid decoherence, they happen we think they happen like 10 megahertz, but then they interfere, and you give slower and slower waves, or eventually getting down to EEG range and cognitive epochs and that sort of thing. So it's a multi-scale hierarchy based on microtubule time crystals that goes inside each neuron, eventually to the Planck scale. But those microtubules in that neuron are entangled with others. So it's actually, you have a global effect throughout wide parts of the brain through entanglement.
Can I ask a question? So the entanglement is not through a biological mechanism; it's a quantum effect, right?
I would call it a quantum biological effect. I think actually entanglement is probably important in biology, but yeah, it's a quantum effect. But I'm saying it's happening in biology.
Yeah.
Uh,
No, the reason I say that is the split brain uh, uh,
yeah, I know
experiments
split
from the
brain.
60s. Yeah. Where they cut the corpus
Corpus callosum.
callosum.
Yeah.
Yeah. And you end up with two separate consciousnesses, or at least two separate uh, hemispheres that appear to be conscious. How would this theory work with that experiment?
Well, first of all, it's unclear whether they actually really severed the two sides because there's a connection anterior, the anterior cingulate cortex. They never connected, so it's unclear whether they're actually truly separated. But I don't see it as a problem. I mean, if you did separate it, then you'd have two brains, and presumably you'd block the entanglement between the two, so you wouldn't have binding. So I don't see that as a problem for our theory.
Okay. Yeah. And can you talk about consciousness, as I said, from the point of view of
Anesthesia.
anesthesia and how
Yeah.
that's different from sleep?
Right. Well, sleep. If you poke somebody when they're sleeping or take a knife to them, they're going to wake up. If you take a knife to somebody who's anesthetized, they're not going to wake up. So it's different. Now, how is it different? Sleep is more hormonal, but it's not the same. The question is, how does anesthesia work? Because anesthesia is selective. Anesthesia takes away consciousness but spares non-conscious brain activities. So, for example, when we do or did spinal surgeries, so let's say they're operating on the spinal cord and the patient's face down, and the back is filleted open, and you want to know whether the surgeon is cutting off blood supply or otherwise screwing with the spinal cord, so the patient would wake up paralyzed. Obviously, we want to avoid that. So what they do is we do neurophysiology, and we have these guys come in, and they put electrodes on the brain and electrodes on the foot or on the hand, and do both motor and sensory evoked potentials. So sensory means you send a stimulus here, the pinky or the hand, and you record it in the other side of the brain. And as long as the spinal cord's working, you're getting those signals. So these guys are watching these signals, and they say,
Mhm.
"Oh, you just pulled too much, lighten up," and then the signal comes back. So these signals are processing, sensory signals are processing, even though the patient is unconscious and doesn't perceive them. And similarly, motor, you can stimulate the motor and cause the hand to move over here, so the signals are going the other way. So you can look at motor. So the brain is actively processing signals without consciousness. So what goes away with anesthesia is a good question, and probably the key to understanding consciousness. So anesthetic gases, there's also propofol and some other drugs, but we'll come to those in a second. But the gases were discovered in the 19th century. Initially they were used for social purposes, for recreation, like ether frolics. If you sniff a little ether, you get high as a kite. You dance around like an idiot. And nitrous oxide, laughing gas. People still abuse nitrous oxide. And virtually, I think any of the anesthetics, if you just take a small amount, you get high from it. And propofol makes you high. Michael Jackson, for example,
Right.
you get about 10,000 times too much, but in low doses, it's tremendously euphoric. So all the gases at low concentrations were euphoric, but at higher concentrations made you unconscious. And they realized that, and they started using it for surgery. And then later, we realized that it's selective, that the evoked potentials and other activities, the EEG is slowed, but it doesn't go away. You still have some EEG. So it affects something. It affects consciousness, but almost little else. Then the paradox is that the gases bind very non-discriminately, non-specifically by quantum forces, quantum interactions, Van der Waals forces. Very, very weak quantum interactions. They don't form chemical bonds. So they just have, so if you have like an aromatic ring, like a benzene or a halogen, full electron clouds, the electrons here are going to repel the electrons here, and these electrons are going to repel these, and they start to oscillate, or they couple. And that's how oscillations work in the microtubules. And the anesthetic comes in, they bind, but they don't oscillate, because it turns out the polarizability is different. So they don't oscillate, and they dampen the oscillation. They dampen the quantum oscillations. So the only thing they're doing is dampening quantum oscillations, and not forming any chemical bonds. So but you have them all over the body in huge amounts in fat stores. I always used to tell the residents, there's more anesthetic in the guy's ass than in his brain, but it's probably working in his brain, don't you think? As a mechanism to point out that there's a lot of anesthesia in the membrane, but that doesn't mean it's working in the membrane. It's actually working in the microtubules, which I thought, but the binding was, but everybody else said, no, it's working on membrane receptors. So eventually it was figured out that anesthetics, despite being lipid soluble, actually work on proteins inside the proteins where the aromatic rings are. And then a question of which protein. So from the mid 80s, and I've been in this field my whole career, so I know this very well. In the mid 80s, it was realized that they act on proteins directly. You don't have to go through the lipid to get to the protein. They work directly on the proteins in the nonpolar interior. And then the question was which proteins. So everybody thought it must be membrane proteins, and they bind the most to GABA receptors. So everybody said it must be the GABA receptors, which are inhibitory. They must potentiate the GABA receptors. Well, the problem is that not all anesthetics bind to GABA receptors, and the Meyer-Overton core. So back I skipped this part. Back around the turn of the 20th century, Meyer and Overton had a bunch of anesthetic gases that had different potencies, but the same potency for every animal. This is actually pretty amazing. All the anesthetics work on all animals and plants, yeah. and for one gas, let's say halothane, its potency is going to be the same for you and I, or for a flea, or for an insect, or for an elephant at equilibrium. So if you allow equilibrium, it takes the same amount to put you or I, or an elephant, or a flea to sleep. Pretty amazing. But that potency would be different from something else. So that tells me that consciousness is the same in all animals. The mechanism is basically the same, and the target has to be the same. And so this became the unitary mechanism. So they thought it was in GABA receptors, but not all anesthetics bind to GABA. And some anesthetics make it open, some closed. There was no consistent picture. So finally, in 2008, after 24 years of studying every damn membrane protein, the leaders in the anesthesia mechanism field, led by a guy named Ted Eager, who was undoubtedly the leader, and they finally gave up. They wrote a paper in main anesthesia journal. Is a new paradigm needed to understand anesthetics? And they said no one membrane protein, or no combination of membrane proteins can account for anesthetic action. And so we need a new paradigm. Well, I wrote and I said, hell yeah, we do. It's microtubules. But they ignored that. They went back to lipids and wasted another 15, 20 years. Beginning in 2006, a couple things happened. Number one, people began to find quantum effects in biology, photosynthesis. For example, photosynthesis used warm temperature quantum biology. But also in the anesthesia world, a lab at Penn, Rod Eckenhoff, began doing systematic. So once they kind of threw out the idea that the GABA was the answer, Eckenhoff's group did a systematic search of all proteins. And for example, they used radio-labeled halothane in a mouse brain, and then took the brain, ground it up, and did gel electrophoresis, and found that 70 different proteins in the mouse brain neuron bound halothane. 70, half in the membrane and half in the cytoplasm. And then they did genomic and proteomic work to find out which protein was functionally involved. And they narrowed it down to the only one that was anywhere involved in signaling was tubulin. And then they did other studies with different anesthetics that showed genomic and proteomic effects from isoflurane and sevoflurane. So there seemed to be genomic, and then they did an optogenetics experiment where they had an anesthetic, an anthracene derivative that was only anesthetic when it was fluorescent, and they gave it to tadpoles who have conveniently transparent heads, and so the tadpoles, they gave them this anthracene anesthetic, which would be anesthetic when it fluoresced, and they were able to swim around and look pretty normal. And they would turn on the UV light, and the tadpoles would go belly up. They'd be anesthetized. And when they ground up their brains, they found that the anthracene was bound to microtubules. And so that's optogenetics. So genomic, proteomic, optogenetic. And then they did a retrospective study on clinical on patients. So there's a drug called Taxol, which binds to microtubules and stabilizes microtubules. And it's used for cancer because the microtubules can't disassemble and become mitotic spindles. So it inhibits mitosis, more so for the tumor cells than for normal cells. So it's an effective anti-cancer drug. So, but it's stabilized. Some of it gets into the blood, gets past the blood-brain barrier. And so they did a retrospective study on patients who on Taxol came to surgery, and looked at their requirements of anesthetic requirements compared to non-Taxol users, and they were significantly higher. So the people on Taxol, which stabilized microtubules and bound to microtubules, needed more anesthetic than did anybody else. And they concluded that anesthetics must destabilize microtubules, or the Taxol is just blocking access to the site, and suggesting that anesthesia works on microtubules. So I reviewed it for anesthesiology. I said, this is a great paper, but they didn't take it. I was probably too excited about it. So they published it somewhere else, but they did publish it. And then recently I reviewed a paper that's going to be coming out pretty soon on where I did a similar thing, but in mice, with a different microtubule stabilizer called apophyllone. Actually, I shouldn't really talk about this because it's accepted in press.
It'll take a while for this to come out. So
okay, so this should come out soon. So basically, they gave it to mice, or to half of the mice, and then did a writing and then they put them in a chamber with anesthetic isoflurane, I think, at 4%, and just waited till they fell over, and that's called a loss of writing reflex. And it was something like 4 minutes on an average. And but the mice on apophyllone required 5 minutes, over 5 minutes, so like a minute longer, pretty long time. And so the paper, I reviewed the paper. I thought this is a great paper. But one of my colleagues, and I know who he was, who was a cartoon neuron proponent, just absolutely ripped it. He just was quibbling over the the weakest shit, and coming up with, no, we can't publish it. I'm going, bullshit. This is a fantastic paper. This is really important. Anyway, I won that argument, and the paper's coming out, and I'll try to remember to send it to you when it comes out. It should come out within a couple of weeks, I think.
Yeah. On a couple of things, just as a little aside, you mentioned plants when you were talking about the effect of anesthesia on microtubules or on consciousness. What are you talking about there?
So plants have microtubules, and a friend of mine named Rajneesh Khanna, who is at the Carnegie Institute of Plant Science at Stanford, did a study where with the well, two things. I mentioned, I'll come back to that. I mentioned my friend Anirban discovering the kilohertz, megahertz, gigahertz, terahertz from the microtubules. It turns out you can measure that from the scalp in humans with a single electrode. You can measure megahertz pretty simply. And you can also, well, Anirban's doing studies in India now with a full DDG, measuring all 12 orders of frequency at various parts of the brain, collecting a huge amount of data that I'm not allowed to talk about yet, but it looks pretty damn promising from our standpoint that there's a change in the megahertz. But you can take a single probe and just measure it, and we messed around with that, and you can see the triplet in megahertz. Anyway, we were doing these experiments, and so we said, let's try it on we tried it on a dog, and we got a nice signal. We tried it on all of us, we got a nice signal. We tried it on an apple and got no signal. And then we tried it on a plant, and I remember Rajneesh had told me that the microtubules are most active at the junction where the root meets the stalk or stem. And so we were looking around. I said, try it down by the stem where the root meets the stem, and there we saw it. We saw a triplet. So he did. So that's where the microtubules control, for example, the turning of the sunflower to follow the sun.
Right.
Well, he did an experiment where he showed that where he put a little isoflurane down in this area by the stem roots, it blocked the turning of the sunflower. So there's that, and so plants put out megahertz, and anesthesia seems to block the turning.
Okay. The other thing, you sort of made a comment, dismissed, not dismissively, but that oh my God, I'm a reductionist. When you were, what isn't this theory of quantum coherence?
Anti-reductionist, because I'm going smaller and smaller. Yes, except when you get to quantum, everything's connected to everything. That's the key. If you didn't have quantum entanglement among all those tubulins, you just have a lot of really smart neurons, but you wouldn't have a smart brain. But if they're entangled and they're acting collectively together, and actually we need 10 to the 15th entangled tubulins in the brain to have a collapse at 10 megahertz. And you could say, well, is that even possible? And the only evidence for biological entanglement, which I'm aware, was done by Anirban between microtubules, or between parameters in microtubules. And what he did was he had a bunch of microtubules, and he irradiated them, and then he separated them. And he had a way of measuring angular momentum in a microtubule. And he found that the angular momentum, when they were separate, the angular momentum of this one was entangled with the angular momentum of this one. My fingers aren't moving the right way, but anyway. So there is entanglement, and now he's doing more and looking at 8 different parameters. So in what we call the quantum underground, where these nonpolar rings are deep inside proteins, I think you can have entanglement. It's a quantum quantum friendly, quantum wonderland.
Yeah,
and they're deep inside.
Right, but why is that anti-materialist? I mean, it's still a physical effect. It's a quantum effect, not a Newtonian effect, but it's still physical.
Yeah, well, usually materialism means classical, strictly classical. And
Okay.
and some people say physical meaning material, but physical can also mean quantum physics. So it's a particular it's quantum physics is interpreted by Roger, which in which collapse occurs through this general quantum gravity or general relativity space-time thing, which connects us to space-time geometry. And that's a key thing because it makes consciousness somewhat fundamental. And all the the Deepak Chopras of the world, and they all say consciousness, and a lot of other people, a lot of physicists now seriously say that consciousness is fundamental, is somehow in tune or attached to the universe. And Roger started all that, although he doesn't like to talk about that. He doesn't
Yeah.
like to talk about anything that would sound woo woo, even though other people might take it that way. And quite frankly, I'm kind of sick of the cartoon neuron people criticizing, because I think what they're saying is the woo woo. That cartoon neurons could give rise to consciousness, I think is
Yeah.
bullshit.
Yeah. It's just when you there are a lot of people at a level much shallower than what you guys are operating at. Think of materialism, dualism, and dualism as pointing to a non-physical realm, and that then bleeds into spirituality and religion and all of that, the existence of a soul. Uh, and so that's what I was saying. Why isn't this reductionist in that the consciousness uh, is a quantum effector emerging from a quantum effect, not from some other non-physical realm?
Well, I'm not sure it isn't. I mean, what do you mean by some other non-physical realm? If you have space-time geometry that is somewhat holographic and repeats at different scales, and if consciousness is a vibration in our process in space-time geometry that can, in fact, go to faster frequencies than can be accommodated in biology, uh, I hate to say it, but consciousness out of the body is not impossible. Until somebody proves that consciousness is a classical computation or classical effect within the brain, we can't rule it out outside of the brain. If it's a quantum effect, you can have quantum non-locality, you can have entanglement between people, you can have parapsychology. It's really a Pandora's box to all kinds of non-local effects, including near death and out of body experiences, end of life brain activity, which has been documented, even afterlife and reincarnation. I mean, you can't rule it out. If consciousness is a process in space time geometry, the door's open to all that stuff.
Yeah.
So I, it's people, they can ridicule it, but we don't know. I mean, you can't say no. I think you can't rule it out. That's for sure, and I think it's quite possible.
Yeah. Okay, we're well over an hour.
Let me just say one more thing.
Yeah.
Two more things. We've done our own experiments uh, showing quantum. We are part of a big study with other theories of consciousness. Our prediction was we would show quantum effects in room temperature and microtubules, and that they would be inhibited by anesthesia. And we showed that, and that was in the Templeton project. So I claim that we have more evidence, far more evidence, a little bit compared to nothing for everybody else, um, for our theory of consciousness compared to the computational theories, which have no evidence, as far as I can tell. They have no explanatory power, no connection to biology, and no experimental evidence. So that's my claim.
What does the future hold for business? Ask 9 experts and get 10 answers. Bull market, bear market, rates rising or falling, inflation going up or down. Can somebody please invent a crystal ball? Until then, over 40,000 enterprises have future-proofed their business with NetSuite by Oracle, the number one cloud ERP, bringing accounting, financial management, inventory, HR into one fluid platform. With one unified business management suite, there's one source of truth, giving you the visibility and control you need to make quick decisions. With real-time insights and forecasting, you're peering into the future with actionable data. If I were a larger organization, this is the product I'd use. Whether your company is earning millions or even hundreds of millions, NetSuite helps you respond to immediate challenges and seize your biggest opportunities. Speaking of opportunities, download the CFO's Guide to AI and Machine Learning at netsuite.com/ionai. That's NetSuite, netsuite.com/ionai, EYEONAI, all run together, to get the CFO's Guide to AI and Machine Learning. The guide is free to you at netsuite.com/ionai, netsuite.com/ionai.